Okay, now we are moving to chapter 4, Essentials of Mendelism. So before this chapter, we discussed the fundamental principles of transmission genetics. Genes are present on homologous chromosomes, so they will segregate for each other. They will assort independently for each other during gamet formation. So it is the basic principles of gene transmission from parent to offspring. So once offspring receives total set of genes, expression will determine phenotype. But when gene expression does not follow the dominant or recessive modes, or when there is more than one pair of genes influences the expression of a single character, usually we will get something that is really different compared to the 3:1 and 9:1 ratio. Elaboration gene function. Genes can exist in alternate forms according to Mendel's experiment. So you have two, dominant and recessive. So this is just a simple dichotomy of alleles, but one allele did nothing, the other allele would determine the phenotype. But research in early 20th century say that it is an oversimplification, because genes can exist in more than two allelic states and each allele can have a different effects on the phenotype. So you have incomplete dominance, codominance, multiple alleles, allelic series, and then we can test the mutation for allelism, and in mutation there is a variation among effects, and the gene function can produce different qualified tests. We can know why are some mutations dominant and other recessive. Dominance. If an allele has the same phenotype heterozygously, just like homozygously, phenotypes BA small a and BA are phenotypically indistinguishable. Sometimes a heterozygote has a phenotype that is not the same as homozygous, dominant, or recombinant, or recessive. For example, flower color in the snapdragon, Antirrhinium majus. So this is a case called incomplete dominance. In Antirrhinium majus, flower color is either red, white, or in between you have pink. The homozygous doesn't create red color just like Mendel, but it becomes a combination between red and white, so it becomes pink. So it depends on the amount of product made by the color gene. So we can say that the red color allele, big W, is not completely dominant. It is incompletely dominant or partially dominant towards the allele for white color, which is small w. If the big W allele specifies this product and the small w allele does not, so big W will have 2 times the product as big W small w heterozygotes do. It will show deeper color. But when the heterophenotype is midway between phenotypes of the two homozygotes, the partially dominant allele is sometimes said to be semi-dominant. Another definition would be codominance. So the human blood type is MN type. One serum anti-M recognizes only the M antigen on the human blood cells. Another serum anti-N recognizes only the N antigen on these cells. So the two alleles appear to contribute equally to the phenotype of the heterozygotes. They found the Nobel laureate Karl Landsteiner. He is the Austrian Catholic and the father of transfusion medicine. The ability to produce the M and N antigens are determined by two alleles from a gene. You have big M, it produces only M antigen. So homozygotes for N will produce only the N antigen, but heterozygotes, they produce both M and N. So M and N will create two proteins at the same time. Neither are suppressed. They are said to be codominant because the two alleles appear to contribute independently to the phenotype. The codominance implies that there is an independence of allele function, meaning that one can function without the other or despite the other. Neither are dominant, neither are recessive towards each other. Multiple alleles. So Mendelian concept states that genes exist in not more than two states, dominant or recessive, but there are actually more alleles for one gene. The Mendelian concept that genes exist in no more than two alleles had to be modified when there is a discovery where genes with 3, 4 or more alleles were discovered. So there are genes with more than two allelic states are common. For example, in the fur color of the rabbit, you have 4 alleles controlling that same phenotype. You have albino, you have Himalayan, you have chinchilla, and you have wild type. Wild type alleles such as C+ is the most frequent allele in the population. Okay, so you can see the 4 alleles: albino, Himalayan, chinchilla, wild type. So if the genotype is c or albino, then the rabbit will be albino. But for Himalayan is CH, like has on extremities. meaning that the whole body is white except for the ears and the legs, it will be black. Chinchilla, you have white hair with black tips on the body, and wild type, you have colored hair over the entire body. So human blood types, or the ABO type. So the gene responsible for producing A and B antigens is denoted by the letter I. So you have three alleles, IA, IB, small I. IA allele specifies the production of the A antigen, the IB allele specifies the production of the B antigen, and the small I allele does not create any antigen. So IA and IB alleles are codominant, but small I allele are recessive towards both IA and IB alleles. So the I gene is polymorphic or having many forms. Blood group B has its highest frequency in Northern Asia and neighboring Central Asia. Incidence diminish both towards the west and the east. So in Europe, it is like 1 or 2 people, single digits. They believe that it has been entirely absent from Native American and Australian Aboriginal populations prior to the arrival of Europeans in those areas, meaning that in those areas, there is very lack of the B blood group. The blood group A is associated with Europeans, especially in Scandinavia, Central Europe, although the highest frequencies occur in some Australian Aborigine populations and the Blackfoot Indians in the Montana in the US. So, the functional relationships among the members of a series of multiple alleles can be studied by making a heterozygous combination. So here, we can actually study which is dominant and recessive towards each other. We call it an allelic series. So there are 4 alleles in the C gene in rabbit, the gene for coat color. So we make 6 different types of heterozygotes. We have C, CH, C, CH, then C, C plus, CH, CCH, then you have CH, C plus, then you have CCH, C plus. So in terms of dominance, the C plus or wild type is the most dominant compared to the chinchilla, compared to the Himalayan, and compared to the albino. So the null or amorphic are the non-functional alleles, which is the albino allele. Hypomorphic are the partially functional alleles, meaning that they are not working as intended 100%. So it is the Himalayan allele and also the chinchilla allele. So to test the gene mutations for allelism, a mutant allele is created when an existing allele changes to a new genetic state in a process called mutation. It's not always possible to assign a new mutation to a gene on the basis of its phenotypic effect. In rabbits, you have several genes determining coat color, and a mutation in one of them will actually reduce, alter, or abolish pigmentation on all the hairs. If there is a new coat color appears, we don't actually know which gene are mutated. Simple tests can be used to determine the allelic identity of a new mutation as long as the new mutation is recessive. The procedure involves crosses to combine the new recessive mutation with the recessive mutation of known genes. Different recessive alleles can be tested for allelism by crossing homologous recessive against each other, since only if they are allelic will the offspring show the recessive phenotype. If they are wild type, then the alleles are in different genes controlling the same phenotype. So this is how we try to look for mutants. First of all, we cross with what we know, and if we cannot find it, they are still wild, meaning that the mutant is actually in another gene that is not the allele that controls the gene that we know. So general scheme is to test recessive mutation for allelism. So two mutations are alleles if a hybrid that contains both of them has the mutant phenotype. It is important to remember that this applies only on recessive mutations. So if you have a hybrid that contains both of them, if they have the mutant phenotype, then you know that is the same allele as the actual original trait. So the mutant mutation cannot be tested this way because they will appear in any type of cross. If you cross it with wild, they will turn up. If you test it with the heterozygous or recessive, they will always turn up. So one such test will be a test on recessive eye color mutations in Drosophila. So you have 3 phenotypically identical mutations, Cinnabar, Scarlet, and Cinnabar 2. We test it for allelism by making pairwise crosses between flies homologous for different mutations. Okay, so let's cross Cinnabar, Scarlet. What do we get? Wild type. Cinnabar 2 and Cinnabar, you get mutant. Cinnabar2 and scarlet will get wild type. So if you get wild type, the cinnabar and scarlet mutations are in different genes. If you get mutant, means that they are alleles of the same gene. Variation among the effects of mutations. Genes are identified by mutations that alter the phenotype in some conspicuous way. For example, color or shape of the eyes, behavior, sterility, and death. So the one that is visible, it affects your morphology or how you look like. Sterile mutation affects reproduction. And lethal mutation disrupt vital functions. So functions which are so important for life, if they're disrupted, then the organism will just die. You can see here, this is an experiment done for the yellow lethal mutation in mice. So there's a cross between two yellow mice to get one grey brown and two yellow. So it seems that you have 2 to 1. It seems that like the yellow is dominant. But if yellow is dominant, shouldn't it be like 3 to 1? Maybe you have one is grey brown, then you have the yellow. Okay, there should be 3 yellow. Instead, what we find is the grey brown is actually double dominant for A+. But for the yellows are always heterozygous. Where did the homozygous yellow go? AY. For AY, they die as embryos. They do not appear in the experiment. So you will always get double number of yellow mice compared to the grey brown. So gene function produces polypeptides. The excessive variation revealed by mutation indicates that organisms contain many different genes. The genes can exist in multiple allelic states, but it doesn't tell us how genes affect the phenotype. If you alter the gene, something happens to the organism, but how? The early geneticists do not have any answer for this question. So, there is a theory called one gene, one polypeptide hypothesis. So, one gene creates a polypeptide, and this polypeptide is needed to make an aspect of phenotype. If one of them fails, then the phenotype will change. Why are some mutations dominant and why are some recessive? Recessive mutations occur when gene loses its function. We call it the loss of function alleles. Dominant mutation occur when a gene acquires new function called the gain of function alleles or the neomorphic allele, which is the new form. Wild-type alleles produces a functional polypeptide. Recessive amorphic loss of function allele does not produce a functional polypeptide, meaning that you have a severe mutant phenotype. But if you have recessive hypomorphic loss of function allele, hypo means low, morphic means shape. So meaning that they are not completely useless, they still have use. It's just that they are kind of weak compared to the normal. It is partially functional polypeptide. So you have a mild mutant phenotype. But if you have dominant negative allele, it produces a polypeptide that interferes with the wild type polypeptide, meaning that this mutation will always happen. We have the severe mutant phenotype. So gene action from genotype to phenotype. Phenotype depends on both environmental and genetic factors. So you have influence of environment and environmental effects on the expression of the human genes, their penetrance and expressivity, gene interactions, efficiency and pleiotropy. So, gene action from genotype to phenotype. At normal culturing temperature, 25 degrees Celsius, Siberian flies are viable and fertile, but extremely sensitive to sudden shock. When the culture is shaken, the flies will temporarily be paralyzed. It will fall to the bottom of the culture. But if we increase temperature to 29 degrees, all the fleas will fall to the bottom and die even without a shock, because the temperature will control how their synapses are sent. Synapses are the messages sent from one nerve cell to a nerve cell. It tells the body how to move. Your brain will use synapses to tell the limbs to flap, to walk, to whatever. But during high temperature, this system just breaks down within the Siberian flies, meaning that they are paralyzed, unable to move when temperature is too high. So as you can see here, 19 degrees, you have the healthy animals. Okay, you can see there's a lot of vesicles. A lot of vesicles carrying, what is it? It carries a lot of neurotransmitters. Neurotransmitters is needed to carry synapses between nerve cell to another nerve cell. But in 29 degrees, you have exocytosis. So there is no more vesicle that carry the neurotransmitter. So meaning that the signals aren't sent from the brain to the legs or to the wings. So they will just fall down. But back to 90 degrees, they will have a full recovery. Gene action from genotype to phenotype. So in humans, we have this thing called PKU or phenylketonuria. Phenylketonuria means that you have phenylketone within your urea, meaning that you are supposed to dump this contents out because it is waste, but somehow it remains in your blood. So because of this, infants who are born with this gene are unable to take out the toxic substances. So you cannot throw out this waste, it remains in your blood, so it will affect your health. So this toxic substance will accumulate in the brain of the baby. It will actually cause the baby to have delayed mental abilities, delayed learning, and also they are unable to communicate. But if the infants are fed with low phenylalanine diet, then they can mature without any problem, because PKU is actually a waste that is created if we eat a lot of phenylalanine in our diet. So whatever we eat, it will be turned to waste. So normally we would just dispose the waste through our urine, but people with PKU are unable to do that. So you have a lot of garbage. Imagine if you have a lot of this food leftover, but you don't have any garbage can or you don't have a dustbin. Soon your house will be full of garbage and your house will stink. So that's how it happens. Your body is unable to dispose of the waste. But what if you do not create the waste at all in the first place? Meaning that if you do not eat any phenylalanine diet, you will not create PKU, then you do not need to deal with the extra toxic substances that is in your body. Environmental effects on the expression of human genes. So for example, is pattern baldness. So the premature pattern baldness is due to an allele that is expressed differently in the two sexes. In males, both homozygotes and heterozygotes will develop bald patches, but in females, only the homozygotes show a tendency to become bald. And the baldness is just a general thinning of the hair. Penetrance. It means that you have the genetics, but you do not show the trait. Genotype is okay, but phenotype doesn't turn up. So you can say that the trait has incomplete penetrance. Specific individuals that show a particular phenotype among those capable of showing it, such as polydactyly. If you look at penetrance, you are born with that gene, but somehow you do not manifest the traits from that gene. If you look at this disease called polydactyly, it is a syndrome where you have more than regular fingers or toes. So some people get it only on the hands, some people get it only on the toes, some people got it hands and toes, some people get only like one digit only, means that one finger, some people got even two fingers, and some of the fingers functional, some of them not functional. If you can see in this picture you can see two hands from the same person. The left hand has an extra thumb, and the right hand has something like a double thumb, meaning that the new thumb is not actually detached from the original thumb, it is kind of together. So in genetics, this is a problem, because in pedigree analysis, you use pedigree to determine where does this disease come from. Is it come from the mother's side or the father's side? But if you have somebody with incomplete penetrance, he will not have the trait. But suddenly when he marries somebody else, and then the children suddenly have the trait. So it is confusing for geneticists because you cannot determine where does the disease come from. Expressivity. It is a trait not uniformly expressed in offspring, but all offspring show the trait, meaning that you have a degree of expression that is differing between individuals. So if a trait is not manifested uniformly among the individuals that show it. For example, if you look at your brothers and sisters, if you have many siblings, you might have a little bit of resemblance to your mother or father, but not exactly. and within your siblings you actually have differences, but you still have the template similar, just like a mom or just like a dad. So if you can see here, this is the Jackson family: Rabbi Jackson, Jackie Jackson, Tito Jackson, Jermaine, Latoya Jackson, Marlon, and then Michael, rest in peace, and then you have Randy and Jane. All of these, they are from the same family, and then you can see the resemblance between Michael, Jackie, Randy even. Okay, right? And you can see they have that expressivity. But if you look at the face, some of them have smaller noses, some of them have big noses, but they look similar. So degree of expression of the trait, it is controlled by a gene. In this case, you can see this is a gene called EYA, or eye absent gene in flies. So this gene controls the expression of eye development in Drosophila. So if the EYA is mutated, what will happen is that the eyes will not be formed in the zygotes of the fly. But there is actually a degree of this expression of the gene. Some of the flies actually have eyes, but the eyes are smaller. Some of them have extra eyes, or we call it ectopic eyes. Some of them have compound eyes, which is gene implication, and some of them have no eyes at all. Some of them even have like small cells that is able to catch light. So this mutation has degrees, so the expressivity is different. Some of the first evidence that trait can be influenced by more than one gene is obtained by Bateson and Punnett. Phenotypes such as high blood pressure are the result of genes with many alleles. So you have an allele for 120 to 80, 100 to 70, and 170 to 95. So high blood pressure is due to interaction from weight, kidney, smoking, and many different factors, not just your genetics. So weight can be controlled by what? By your obesity genes. Kidney function controlled by your salt repository genes, because if you have too much salt, you have high blood pressure. Smoking, it is controlled by a gene for addiction tendency. All of these mismatched together, you can actually have a risk for high blood pressure. Gene interactions. R and P are both genes that control a trait called the comb in the roosters. In the roosters, or in hens, in chickens in general, you have four types of combs. One is the rose comb, one is the pea, one is the walnut, and you have single leghorns. The distribution is not following Mendel at all. All of these genes, they create the comb, but as the degree of expression is very different. If you have both dominant big R, big P, you have walnut. If you have big R and big P, small p, if the P is heterozygous, you still have walnut. What about the second one, rose? Okay, so rose, we have big R, small p. Mean that as long as the small p is there, you will have the rose. What about P? So for P, or Brahmas, the R must be recessive. Small r, big P. R must be recessive, but P must be dominant. What about the single, the leghorn? For leghorn, both must be recessive. Small r, small p. Epistasis. It is an important genetic phenomenon involving two or more genes influencing a single trait. An epistatic gene is when an allele of one gene controls or overrides the effects of the others. Many genes affect the synthesis of eye pigments. Many genes affect synthesis of eye pigments in Drosophila, and flies homozygous for any alleles have abnormal eye color. Okay, so as you can see here, to make the normal color of the fly, you actually need two genes to be turned on, right? You have the gene for drosopterin and gene for xanthomatin. But controlling both of these genes, both of these genes depend on a pigment transporter. Mean that without the pigment transporter, both colors will not be created. So when two or more genes influence a trait, an allele of one of them may have an overriding effect on the phenotype. So if you look at this picture, you have white and purple flowers on sweet pea. So this is the interaction between products of non-allergens. Okay, so sweet peas. Two flower color loci in the sweet pea, C and P, encode enzymes that synthesize anthocyanin in two sequential steps. So mean that you have step 1, step 2. Okay, so if step 1 and step 2 are not fulfilled, the color purple will not be created. If both of the alleles are homozygous, the pigment will not be produced. So if you can see there, big C, small p, cross with small c, big P. The gametes you will have is big C, small p, and small c, big P. So F1, all of them are heterozygous. Big C, small c, big P, small p. Purple is big C, small c, big P, small p. Alright. So now, when we cross them all together in this punnet, you can see that the one that has big C and big P, both of them dominant, they become purple. If they have one recessive allele or recessive trait, they will be white. So both of them must be dominant for it to become purple. So you get 9 to 7. 9 purple, 7 white. This is another example called the shepherd's purse plant. The shepherd's purse plant is also controlled by two genes. The two genes can act to produce triangular shape, control triangular or ovoid seed capsules in the shepherd's purse. So they both act in the same step in parallel, so that ovoid capsule only occur in double recessive homozygotes, leading a 15 to 1 ratio when double heterozygotes are crossed. Dominant alleles of one gene is epistatic over the recessive allele of the other. So it doesn't follow 9:21, but it follows 15 to 1. In a nutshell, this is how it is. You have a precursor, they have gene A, gene B to get product. The product will form a phenotype. So it means that if A is turned on, you can still have the product. If B turned on, you still have the product. The only way to not get the product is both genes are off or recessive. So if you have big A, big B, triangular, because you still have product. Small a, big B, still triangular, because you still have gene B will create the product. And then you have big A, small b, you will still become triangular because you still have the gene A that will create the product. And then you have small b, mean that none of the genes are actually dominant, mean that they will not create the product. They will become ovoid. Precursor, yellow pigment, green pigment. So in this case, the color is controlled by two alleles. Both are working to make the pigment. But before getting the color, you must get the precursor to be done first. So if you only have the C genes or C alleles dominant, you only get yellow. But if you get C and G both dominant, then you get green. If both C and G are turned off or recessive, you won't get any color at all, because yellow is gone, green is gone. So you have big C, big G, you get white. Big C, small d, white. Big C, small c, G, yellow, because only the yellow ones turned on. You have big C, small d, you get green. Pleiotrophy. It is a condition in which a single gene influences more than one trait. For example, phenylketonuria and singed bristle mutant in Drosophila. Singed is a burnt appearance. PKU in humans, and not just control one trait, it controls a lot. It just doesn't just dump a lot of this waste in your blood, which is phenylketonuria. It also alters your skin and hair color besides mental impairment. So in phenylketonuria, you have mental impairment interfere with the synthesis of melanin pigment, and it lightens the color of the hair. So in this case, this waste called phenylketone, it will just stay in your blood. If it reaches your brain, it makes you a little bit low IQ. It interferes with synthesis of melanin pigment, in that the color of your skin will not be created. So it actually causes the hair color to become light. Mendel's 7 traits were controlled by alleles acting discontinuously. For example, tall or dwarf, yellow or green. But many traits such as height, weight, and agricultural yield are complex and are controlled by many genes, so they can vary continuously within a population. Each gene contributes a small effect to the trait, so Mendelian segregation is virtually impossible to demonstrate without the statistical method developed by quantitative genetics. Onset of genetic expression. Not all genetic traits become apparent at the same time during an organism's lifespan. In most cases, the age at which mutant gene exerts a noticeable phenotype depends on events during normal sequence of growth and development. In humans, the prenatal, infant, pre-adult, and adult phases require different genetic information. As a result, many severe inherited disorders are not manifested until after birth. For such disease, this disease happens in the Jewish community. So newborns appear to be healthy when they are born. After a few months, suddenly it hit them like a brick. So they start to become developmentally retarded, paralyzed, and blind. And then a lot of these babies with such disease will not live until the age of 3. Nash-Nyhan syndrome. It is inherited as an X-linked recessive disease. Abnormal nucleic acid metabolism means that just like the PKU, you are unable to take out the waste from your diet. So what happens that a lot of these nucleic acid, they will just stay in your blood, and then it will become nitrogenous purine bases. And then these nitrogenous purine bases, they become uric acid. And as uric acid, this is actually something that you do not want in your body at all. You want them gone, but you know, you got trash in your apartment, you got trash in your home. What you want to do? It actually causes a lot of bad things such as uric acid in blood, the tissue, mental degradation, palsy, septalization in the lips and fingers. Mean that people with this disease, they keep biting their fingers, biting their nails, okay, because they feel the itch, so they mutilate themselves. It is caused by a mutation in the gene coding hypoxanthine guanine phosphoribosyltransferase transferase. Means that this is an enzyme called the phosphoribosyltransferase transferase to kind of like to transfer the different parts of nucleic acid. This gene is actually broken, so your body could not create that enzyme to break down the nucleic acid. So this disease also is similar to the Tay-Sachs disease. The baby is born healthy, but after 6 to 8 months, suddenly they become afflicted with the disease. Huntington disease. This is the most delayed and highly variable age of onset for genetic disorder in humans, and also autosomal dominant. So in Huntington disease, your frontal lobes of your cerebral cortex will start to degrade, and that you have progressive cell death that occurs in over 10 years. If you look at the picture on the right, the human brain in a normal condition is the one below. After Huntington, you can see that the brain has degenerated. I mean that there's lack of volume in the brain, and then the sulcus or the ridges actually become bigger and more farther apart, meaning that the brain has lost a lot of the mass. So brain deterioration is accompanied by spastic and controlled movement, intellectual deterioration, and untimely death. Onset of these symptoms reported at all ages, but most often initially observed at ages 30 to 50. The mean age of onset will be 38 years. So mean that you are born with the genetics, but the disease doesn't happen until you are 38. Then it kicks in. So gene products may play more essential roles during a certain time during an organism's life cycle. So in the impact of mutant gene can be tolerable for a period of time without noticeable effect, but at one point the mutant phenotype is manifested. At one point, a mutant phenotype is manifested. It may be the result of internal physiological environment of an organism changing during development and with age. Hereditary anticipation. Heritable disorder that happen at an earlier age compared to what is expected, and more severe in each successive generation. For example, DM, myotonic dystrophy, the most common type of adult muscular dystrophy. Individuals affected with this autosomal dominant disorder exhibit extreme variation in the severity of symptoms. Some people get it light, some people get it medium, some people get it heavy, some people cannot even live because their muscles just fail. If it is mild, usually you'll have cataracts as adults, but have very little or no muscular weakness. Severely affected individuals would have more extensive weakness and myotonia, which is your muscle would hyper-excitable. Okay. In some cases, mental retardation, but the more extreme form is fatal just after birth. So in 1989, this research done by C.J. Howler studied 61 parent-child pairs. 60 pairs of the cases age of onset were earlier and more severe in the child than his or her affected parent. So in this picture, you can see the grandfather had it at 55 years old. One generation later, the daughter get it in 50 years old. The son get it in 48 years old. And then the grandson get it in 41 years old. Okay, as you can see, there's a gap. The grandfather got it during the age of 55, but the grandson got it in a younger age, 41 years old. So 14 years old earlier than expected. So, is there a molecular cause of the mutation? So, there is this 3 nucleotide DNA sequence of the DM gene repeated a variable number of times, and it is unstable. Normal individual would have 5 to 35 copies of the sequence, but affected individuals would have between 80 and more than 2,500 copies. The more number of repeats, the more severely that you are affected. In successive generation of DM individuals, size of the repeats increases. So, as the generation goes on, more repeats, more severity, and earlier onset. Genomic parental imprinting and gene silencing. So, the process of selective gene silencing occurs during early development, impacting on subsequent phenotypic expression. For example, you have genes or regions of chromosome are imprinted in one homolog but not the other. Impact depends on the parental origin of the genes and region involved. Silencing leads to the direct phenotypic expression of the alleles on the homolog that is not silenced. Thought to occur before or during gamete formation, leading to differentially marked in genes or chromosome region in sperm forming versus egg forming tissues. Example in humans, you have 2 distinct genetic disorders thought to be caused by differential imprinting of the same region of the long arm of chromosome 15q1. So in both cases, disorder is due to an identical deletion of this region in one member of the chromosome 15 pair. So every one of you would have 2 chromosomes, one from the mother, one from your father. So in this case, you have chromosome number 15. So if the chromosome number 15 from the mother is disabled or deleted, you have one disease. But if you have same thing happening to the other chromosome, mean that your father's chromosome, mean that you only have one chromosome working for your mother, so you have another disease that is different. So Prader-Willi syndrome, PWS. So it happens when the paternal segment is deleted. The one that remain are the undeleted maternal chromosome. So if the maternal segment is deleted and an undeleted paternal chromosome remains, we have an entirely different disorder, which is Angelman syndrome. For PWS, you have mental retardation, a severe eating disorder marked by uncontrollable appetite, obesity, diabetes, and retardation. And then you have Angelman syndrome, you have mental retardation by involuntary muscle contractions, which is called chorea, and seizures. So Angelman syndromes, people with Angelman syndrome, they will usually be unable to learn, unable to emote. The only thing that they know is to laugh. They laugh all the time, and they cannot even walk, and they cannot even comprehend what is happening around them. But if you compare it with PWS, Prader-Willi syndrome, their intelligence are kind of like the Down syndrome people. So they can understand language, they can listen to whatever you say, they can even communicate their thoughts. It's just that they feel hungry all the time. Conclusion: the evolved region of chromosome 15 is imprinted differently in male and female gametes. Both the undeleted maternal and paternal regions are required for normal development. So you need one for your mother, one for your father. Both of them are needed so that you can live naturally. If you lose one part of the mother's side or the father's side, you will have a disease, either Angelman or Prader-Willi. Multiple allelic inheritance. So you have the Bombay phenotype. So in 1952, there is a very unusual situation provided information concerning the genetic basis of the H substance. So a woman in Bombay, she has the blood type which is O, seemingly O because her blood didn't react to A or B antigen. So people assume that her blood was O. But however, one of her parents were type AB, and she herself was an obvious donor of an IB allele for 2 of her offspring. Meaning that because her parents are having an AB, right, and she gave her son a B, even though she married a man who is A, so the B comes from her, meaning that she was genetically type B but functionally type O. This woman was subsequently to be shown as homozygous for rare recessive mutation in the gene designated FUT1. So it is the gene fucosyltransferase. In this mutation, terminal portion of the carbohydrate chain protruding from the red cell membrane lacks fucose, normally added by the enzyme. In the absence of fucose, the enzymes specified by IA and IBLs apparently are unable to recognize incomplete H substance as a proper substrate. Thus, neither terminal galactose or N-acetyl galactosamine can be added, even though the appropriate enzymes capable of doing so are present and functional. Meaning that they are unable to recognize the incomplete H substance, they will not be assigning any types for the blood. But the blood will continue becoming an O blood instead of an A or B blood. So, with the mutant in FUT1 gene, as a result, the ABL system genotype cannot be expressed in individuals homozygous for the mutant form of FUT1 gene. Even though they may have the RA or IB alleles, neither gene is added to the cell surface. So fortunately, they are type O. To distinguish them from the rest of the population, they are said to demonstrate the Bombay phenotype. The frequency of the mutant FUT1 allele is exceedingly low. The vast majority of the human population can synthesize the H substance. It means that in the Bombay blood case, you can see this is a clear marker of epistasis. You have the gene that controls your type A, B, AB, and O, but all of these genes rely on the FUT1, which enables blood to have a type. If you do not have the FUT gene working well, you won't even have a type at all. Okay, so individuals with a genotype with A or B allele would not be able to express it because they don't have the FUT1 gene. If many individuals with FUT mutation have children, you will have 3A, 6AB, 3B, and 4O. What happened? Because 4 of these children would not have their own blood type. In Drosophila, you have the white locus. In Drosophila, there are many alleles that are present at practically every locus. The recessive mutation that causes white eyes are discovered by Thomas H. Morgan and Harvard Bridges in 1912. It's one of over 100 alleles that occupy this locus. Eye colors range from no pigment white allele to deep ruby in white Satsuma allele, orange in white Albrecht allele, and buff color in white buff allele. So, in this locus for white, you have many alleles, W, WA, WBF, WPL, all the way until white tinged, which is light pink. So the total number of pigment in these mutant eyes is reduced to less than 20% of what is found in the red wild type. So the product of the white locus is a protein that is involved in transporting pigment to the omatida, which is, the eyes of the fly are actually compound eyes. There are so many small eyes. They are combining together to make a large image for the retina. Imagine like you have many cameras, okay, and so omatida is one of those small cameras on the eyes. Okay, so while flies expressing the white mutation can synthesize eye pigments normally, but they cannot transport it into the structural units of the eye. I mean that these flies, they actually can create that color, red color, yellow color, okay, or deep red, just like the wild type, but they are unable to move that color to the eyes. So the white eye phenotype is rendered. These are the allele represent essential genes. So many gene products are essential to an organism's survival. Mutation resulting in synthesis of gene product that is non-functional can be tolerated in the heterozygous state because one wild-type allele can produce enough essential products to ensure survival. However, homozygous recessive will not survive because in nature, some of the alleles they actually interact in different ways, not just like Mendel's dominant recessive. Some of them can actually bring out new possible phenotypes. Okay, for example, summer squash. If you cross a plant with disc-shaped fruit, big A, big A, big B, big B, with plant with long fruit, small a, small B, small b, every generation will all be disc fruit. Mean that you know that A and B, case B, B are actually dominant, but in F2 progeny, the fruit with a new shape, sphere, round like a ball, they appear as well as fruit exhibiting the parental phenotypes. So both gene pairs will influence the fruit shape equally. Your dominant allele of either locus ensures a sphere-shaped fruit. In the absence of dominant allele, the fruit is long. But if both of them are present, it becomes a disc shape. So inheritance of eye color in Drosophila melanogaster. The wild-type eye color is brick red. When two autosomal recessive mutants, brown and scarlet, are crossed, the F1 generation consists of flies with a wild-type eye color. In F2 generation, wild scarlet, brown and white-eyed flies are found in 9:1 ratio. So wild-type eye color is due to the position and mixing of two separate pigment groups, bright red drosophila and brown xanthomathins. Each subpigment is produced by a separate biosynthetic pathway. In the presence of at least one wild-type BW plus allele, an enzyme is produced that converts substance B to C, and the pigment of drosophila is synthesized. In the presence of at least one wild-type ST plus allele, the substance E is converted to F, and the pigment of xanthomathin is synthesized. So, as you can see, to actually create each one of the pigments, you need something else. You need a prerequisite. If you have a mutation, then the pathway to make either one of these colors or one of these pigments will be blocked. So to make the wild-type eye color, you need brown and red. If you have ST mutation, you will not have any xanthomathin, no brown, so you only have red. So you have a scarlet mutant. What if you have a mutation in the BW? So you block drosophila, no red, but you have brown. So you have a brown mutant. But if you have a mutation in both of the alleles, the drosophila and xanthomathin, both of those are not created, then you have a double mutant, which is no color at all, white. The end.